So participants, I have launched the technical poll. Quickly answer me. My audio is clear and my screen is visible, so that we can proceed with the lecture. Okay, okay. Hello everyone. Today we are going to discuss biomolecules. We'll do a quick revision of biomolecules. Along with that, we also do the previous year questions as well. As you all know, what is biomolecules? Like, as the name suggests, bio is living. So all the molecules which are present in a living organism, those molecules are known as biomolecules. So two analysis has been done. One is the chemical analysis and the second one is the ash analysis. First one is the chemical analysis and the second one, this is your ash analysis. So in chemical analysis, what the scientists did, they take the living tissue. It can either be a plant tissue or an animal tissue. So they take that living tissue and they grind it in a with the help of mortar and pestle. After grinding for grinding, they use a chemical which is a trichloroacetic acid. This one. So they take the living tissue, mix the trichloroacetic acid and grind it with the help of mortar and pestle. They get a slurry. When they filter the slurry, let me make a funnel tube. This is a beaker. They put this slurry. They use cotton or cheesecloth for filter. And this is a filtrate. They observe two components. One is the filtrate. And the second component is retentate. Okay. So the molecules which are smaller in size, they can easily dissolve in acid. So filtrate consists of acid soluble pool. The molecules which are smaller in size or micromolecules. And the molecules which are larger in size, they are not able to dissolve or soluble in pool. It consists of acid insoluble pool. So all the macromolecules, because they are larger in size, they come in acid insoluble pool, your polysaccharides, nucleic acid, proteins, carbohydrates. Okay, they all come in acid insoluble pool. Second analysis, the ash analysis. What they did, they took the living tissue, they dry it and they burnt it. So the organic component like carbon remove in the form of carbon dioxide. But the ash which is left after burning, this ash contains the inorganic elements which are calcium and magnesium. Okay, I hope this is clear. Next one is the metabolites. So there are two types of metabolites. One is the primary metabolite and the second one is secondary metabolites. So primary metabolites are those which play a very important role in the main chemical reactions or which are required for the normal physiological reactions which takes place in our body. Okay, secondary metabolites are those metabolites which are not essential for the main metabolic reactions. Like for example, the examples of primary metabolites are sugars, proteins, right? And the example of secondary metabolites are carotenoids, morphine, codeine, alkaloids, essential oils. So secondary metabolites are the byproducts. And this table, it's very important. It is given in NCERT, very, very important. Please learn this table by heart. Okay, moving to the next one. In the next one, we're going to discuss the protein. All you know that proteins have a monomer. That monomeric unit is amino acid. And these amino acids, they are joined with a bond, and that bond is a peptide bond. And when more amino acids are like 1, 2, 3, 4, 4, 5, 6, 7, like many amino acids are joined together by this peptide bond, they form a polypeptide chain, and that polypeptide chain is known as protein. Monomeric unit is amino acid. Protein is a heteropolymer. Remember this, students. Protein is a heteropolymer, not a homopolymer. There are some essential amino acid and non-essential amino acid as well. Essential amino acids are those amino acids which are not synthesized in our body. We have to take this amino acid from outside, like from food. And non-essential amino acids are those which can easily synthesize in our body. Collagen is also a protein. So collagen is an abundant protein in animals. If the question says, like, name the protein which is abundant in animals, then the answer would be collagen. But if they ask you, name the protein which is, okay, if they ask you, like, name the protein which is abundant in the biosphere, then you have to answer rubisco. If they ask you about the biosphere, then you have to answer rubisco. Based on the number of carboxylic group and amino acid, on the amino group, the amino acid can be like acidic, basic, neutral, and aromatic. Aromatic when they have in ring. Okay. Acid amino acid when they have a more carboxylic group as compared to the amino group. Basic when they have more amino group as compared to carboxylic group. Like example of basic is lysine. Acidic amino acid is glutamic acid. Neutral is valine. And Zwitter ion when the amino acid exists as a bipolar ion in solution, when we dissolve it in water, like plus, minus, they have both the charges, dipolar. Moving to next one. This slide shows you the structures of protein, like primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure, when the amino acids, they are like primary structure, so it shows you the sequence of amino acid. Amino acid, they are arranged in a chain, right? A polypeptide chain. And the secondary structure shows you the helix and the beta sheets. Tertiary structure, which gives you a very unique three-dimensional conformation of the protein. And this tertiary structure, very important for the biological activity of the enzyme. Remember this point. Which structure is responsible for the biological activity of enzyme is the tertiary structure. And the quaternary structure, when there's more polypeptides subunits are present. This table shows you the functions of protein. There's a protein like collagen is a protein, function is intercellular ground substance. Trypsin is a protein, it functions as an enzyme, right, in digestive process. Insulin is a protein, it functions as a hormone. Like insulin lowers the blood, lowers the sugar in the blood, right? Antibodies, they are the protein and they fight against infection. Receptor is also a protein, like sensory reception, smell, taste, hormones, etc. This one, GLUT4 is a glucose transporter. It allows the glucose to transport into cell. Moving to the next one is the carbohydrate. So carbohydrate, they are polyhydroxy ketose or aldose. So carbohydrate, there are of major classes are four. One is the monosaccharide. They have glucose and fructose. Disaccharides, that means that has two units. Remember, di means two, mono means one. So monosaccharide examples are glucose and fructose. Disaccharide, there's sucrose, lactose, and maltose. Like sucrose is consists of two units. One is the glucose and second one is the fructose. And remember, the sucrose is a non-reducing sugar. Remember this point also. Is a non-reducing sugar. If I say non-reducing, that means the reactive group is not present at the end, or the reactive group is not free. Okay. Oligosaccharide is raffinose. Polysaccharides, when many monomers, like when monosaccharides unit, they join together and they form a very long chain. These are polysaccharides. Like example is cellulose, starch, and glycogen. Starch is consists of two units. One is amylose and second one is amylopectin. Okay. So amylose is unbranched. Remember this. Amylose is unbranched and it shows 1,4 alpha 1,4 glycosidic linkage. Amylopectin is branched. And it shows alpha 1,4 plus alpha 1,6 glycosidic linkage. Okay. Now next come to the cellulose. Cellulose is unbranched. Remember this. And the glycosidic bond is beta 1,4 glycosidic linkage. Glycogen is branched. Remember all these points, beta. Next one is ilunin. Ilunin is a polymer of fructose. Remember this, it's very important. Like cellulose is a polymer of glucose. Starch is also having a glucose. Glycogen is also having a glucose, but inulin is a polymer of fructose. Star shows iodine test because of this helical structure, because of these presence of because of the presence of the helical structure, the iodine get trapped in this helical structure and cannot move out. Because of that, the starch gives a blue-black color. It shows positive iodine test. Glycogen, the left end is non-reducing and the right end is reducing. As I told you, non-reducing end when the reactive group is not present or the reactive group is not free. Reducing end when there is a presence of a free reactive group. Cellulose is a polymer of glucose, just I told you. I'm moving very fast, beta, because we have to cover a lot of things. Next one is a nucleic acid. Nucleic acid are of two types, like DNA and RNA. DNA, when they have a deoxyribose sugar, and RNA when they have ribose sugar. Deoxy, at the second position, hydrogen is present. Instead of hydroxyl group, hydrogen is present. Okay. The monomeric unit of nucleic acid is a nucleotide. Nucleotide, it consists of sugar, it consists of phosphate, plus it consists of nitrogenous base. And if only sugar and nitrogenous base are present, then they are known as nucleoside. Remember this, if sugar and nitrogenous base are present only, then it is a nucleoside. And if phosphate, sugar, nitrogenous base all are present, then it is a nucleotide. Okay, nitrogenous base are of two types. One is purine and the other one is pyrimidine. Purine, that double ring. In pyrimidine, the single ring. Like adenine, guanine, they are example of purine, and pyrimidine is cytosine, thymine, uracil. Okay, you can see that there's a sugar, the phosphate. These are your nitrogenous bases. The adenine is binding with thymine with a 2 hydrogen bond. 1, 2, 2 hydrogen bond. Here, the guanine is binding with cytosine with a 3 hydrogen bond. Remember this. Adenine binds with thymine with a 2 hydrogen bond, and guanine binds with cytosine with a 3 hydrogen bond. Okay, and the sugar, the phosphate group, this bond and this bond, they are the phosphodiester bond. Sugar and nitrogenous bond, this is a glycosidic bond. And the bond between the two nitrogenous bases is the hydrogen bond. Remember this. Okay, moving to the next one. Lipid. Lipid is water insoluble and it's not a polymer. This is a very important point. Remember, note this down. That lipid is not a polymer. Simplest form of lipid is fatty acids. And the fatty acid consists of long chain of hydrocarbon and one carboxylic group. This is the carboxylic group, and this is a long chain of hydrocarbon. So fatty acid could be a saturated and unsaturated. Saturated when they have a only single bond. Unsaturated when they have double bond. Remember this. Okay, and the fatty acids are amphipathic in nature. Amphi means that they have both polar plus non-polar. Remember plasma membrane? The plasma membrane is phosphobilipid. It's a phosphobilipid. It has polar head and a non-polar tail. So it has both polar and non-polar parts. So it is amphipathic in nature. Ganglion oil has low melting point. Remember this. Okay, I hope this is clear. And one more point beta regarding lipid. The lipid are not strictly macromolecules. Why? Because lipid have a size which is smaller or less than 800 Dalton. Okay, but they are still considered as macromolecule. Why? Because they show that they show the presence in an acid insoluble pool, right? Lecithin beta. It's lecithin. Okay, I hope it is clear to everyone. Okay, we're moving to the next slide. Enzyme, very important. Enzyme, almost all enzyme are protein. Almost all enzyme are protein, but some nucleic acid behave as enzyme, and those are ribozyme. Those are ribozyme. So, enzyme are the organic catalyst. And they're inorganic catalysts as well. And these inorganic catalysts, they work efficiently at high temperature and high pressure. But the enzyme, they cannot work at very high temperature. They become denatured because enzymes are mostly proteins and the protein get denatured at high temperature. But in organic catalysts, they work efficiently at high temperature as well as at high pressure. But if we isolate the enzyme from the organism which are living in a very high temperature, like 80 to 90 degrees Celsius, those enzymes which are isolated from that organism, those enzymes will be thermostable. Okay, like Taq polymerase. This is an enzyme which is used in PCR, and it is a thermostable enzyme. And this enzyme is obtained from the Thermus aquaticus. Okay. Now let's come to this figure. This is an enzyme and this is a substrate. Okay. Each enzyme has an active site, the site where the substrate will bind. And this site is specific, like a specific substrate can bind to a specific enzyme. Okay. So this site is highly specific. When the substrate bind to the enzyme, they form a complex, and this complex is known as an enzyme substrate complex. And this complex is for a very short duration. Okay, this enzyme substrate complex is for a very short duration, And this enzyme convert the substrate into product and a free enzyme, and we will get a free enzyme. Now this enzyme is again ready to bind to some other substrate. Okay. And metabolism. Beta, metabolism is a sum total of all the chemical reactions which are taking place in our body, like all the reactions. It can be a cellular respiration or the Krebs cycle which is taking place in mitochondria, like all the sum total of all the chemical reaction which is taking place in a living organism, that are metabolism. Metabolism is of two types, there's catabolism and anabolism. Catabolism, when a complex molecule is broke down into a simpler molecule. When a complex is converted into a simpler, and in this process, the energy is released. Why the energy is released? Because the bonds are break. When the bonds break, the energy is released. Anabolism, when simpler molecule form a complex. When all the simpler molecules, they join together and they form a complex molecule. In this process, the energy is absorbed. So it's a endothermic process in this, because in this process the bond formation will take place, and bond formation will require energy. And also remember one point, that all the living state, okay, that a living state is a non-equilibrium steady state able to perform work. If I say that a living state is always in non-equilibrium. And a steady state. Steady states, I mean that is constant. Like every molecule has a fixed concentration, like glucose in the blood have a concentration of 4.5 to 5 millimole. They have a fixed concentration in the blood. If the concentration of glucose will increase in the blood, it will lead to a diabetes, right? So every molecule has a fixed concentration. Okay. And you remember that glucose is converted to pyruvate during cellular respiration process, and the pyruvate is converted to acetyl-CoA. Then acetyl-CoA will enter into the Krebs cycle, where NADH, FADH2, CO2, and ATP are formed, right? And then NADH and FADH2, they will move to the electron transport chain, and finally you get the ATP synthesis, right? So in equilibrium, what happens generally? If A molecule is converted to B, the B will convert again to A. This is what we call equilibrium. Like A is converted to B and the B is again converted to A. But in living state, this reaction does not take place. The equilibrium is not occur in the living state, because glucose is converted to pyruvate. If what will happen if pyruvate again will convert back to glucose? Then you won't be able to get acetyl-CoA, you won't be able to get like any Krebs cycle, there will be no Krebs cycle, no ATP, right? So a living state shows non-equilibrium. So a living state shows non-equilibrium and a steady state. I hope this is clear. This line is given in bold in NCERT. Moving to next is rate of reaction. As you know, that the enzyme, they catalyze the reaction, they increase the rate of the reaction by lowering the activation energy. The activation energy is the energy which is required to start the reaction. So if you lower the activation energy, the rate of the reaction will get double, or it will increase the rate of the reaction. This is a substrate, right? This shows the energy with enzyme, and this shows the activation energy without enzyme. You can easily make out the difference. The enzyme has lowered the activation energy, right? If the product lower than substrate, in this case, the product is lower than the substrate, that means the energy is released. The reaction would be exothermic. And if the product is higher than substrate, the energy will be absorbed. Okay? I hope this is clear. And there are many factors which affect the enzyme. One is the pH, then temperature, substrate, concentration, and inhibitor as well. First, let's discuss the pH. We get this bell-shaped curve. Here's the enzyme activity. This is the pH. This is the point, the optimum point, optimum pH where the enzyme activity is maximum, where you see the maximum enzyme activity at a optimum pH. If I increase the pH or if I decrease the pH of the reaction, the enzyme activity will decrease, because the enzyme, they show I would say a increase in the rate of reaction only at an optimum pH. Okay, remember this point. You can see that this pH, the enzyme activity is maximum. And if I increase the pH, the enzyme activity will decrease. You can see that it is declining. Okay, similarly goes with temperature as well. It also shows the bell-shaped curve. At optimum temperature, the enzyme activity is maximum, and if I decrease the temperature or increase the temperature, it will affect the enzyme activity. The enzyme activity will decrease. Now the substrate concentration. If I increase the concentration of substrate, the enzyme activity will increase, but up to a certain point. Okay, it will increase to a certain point. After some point, if I keep on increasing the substrate concentration, the enzyme activity will not increase. Why? Because of all the enzyme will get saturated. Now the enzyme, they don't have the free active site where the substrate can bind. Even if there's a concentration of substrate is more in the solution, but still the enzyme will not be able to get bind because they don't have a free active site. All the enzymes, they are occupied, all the enzymes are saturated. Okay, so the substrate, if I increase the substrate concentration to a certain limit, it will show the increase in the rate of reaction, but to a certain limit. After that, if I keep on increasing the substrate concentration, the rate of the reaction will not increase. Other factor is an inhibitor. Malonate. Remember everybody? Malonate. Malonate is a competitive inhibitor of substrate succinic. How should I write it? Succinate. Okay. And the enzyme which malonate inhibit is succinic dehydrogenase. Okay. So competitive inhibitor are those inhibitor which mimics the structure of the substrate. Like the structure of the substrate as well as the inhibitor are same. Okay, so there's a competition, as the name suggests, competitive. So there's a competition between the substrate and the inhibitor for the active site of the enzyme. Moving to next one is the classification of enzymes. So enzymes are classified as oxidoreductase, there's transferase, hydrolyase, lyase, isomerase, and ligases. Oxidoreductase, which catalyze the oxidoreductase reaction between the two substrate. Example is peroxidase. Transferase, which catalyze the transfer of a group between a pair of substrate. Example is kinase. Hydrolysis catalyzes catalyzing the hydrolyzed of ester, ether, peptides, glycosidic bond, and the example is peptidases. Similarly, lyases, they catalyzed or removed the group from the substrate by mechanism other than hydrolysis. Remember this. And the example is aldolases. Isomerase, they catalyze the interconversion of optical, geometric, or positional isomerase. And the isomers, and the example is epimerases. Ligases, they catalyze the linking together of two compounds. Example is carboxylases. Okay, remember all the classes of the enzyme. It is given in NCERT. Okay. Now let's come to cofactor. As you know that enzyme has two parts. One is the protein part, we call it as apoenzyme. And the second one is the non-protein part, and we call this non-protein part a cofactor. These cofactors are of three types. They can be a prosthetic group, coenzyme, or the metal ions. Okay, so cofactor plus the apoenzyme, which is the protein part of the enzyme, they will result into a holoenzyme. And this holoenzyme is biologically active. Okay, remember the apoenzyme alone, it is biologically inactive. Okay, and once the apoenzyme binds with the cofactor, which is a non-protein part, then it will lead to a formation of a holoenzyme, and that holoenzyme is biologically active. So cofactors are of three types. One is the prosthetic group. Prosthetic group where the organic compounds that tightly binds to the protein part of the enzyme, that is apoenzyme. Example is heme group in peroxidase and catalase. Second was the coenzyme. Coenzyme when the organic compounds are temporarily bound to apoenzyme. Okay, and the metal ions, they show the coordination. They show a coordination of bond at the side chain of the active site and with the substrate as well. So the coordination bond is formed between the side chain at the active site of the enzyme and also at the same time with the substrate itself. Metal ion, they form the coordination bond with the side chain at the active site of the enzyme and at the same time with the substrate. Example of metal ions are zinc. Zinc is a cofactor for the proteolytic enzyme carboxypeptidase. And the example of coenzyme is NAD. Remember that NAD and NADP contain vitamin, which is niacin. Okay, and this NAD and NADP are coenzyme. I hope everything is clear. Moving to next is the question. Question says, match list 1 with list 2. In list 1, protein A is protein, B is unsaturated fatty acid, C is nucleic acid, D is polysaccharide. List 2 says 1 is CC double bond, 2 is phosphodiester bond, 3 glycosidic bond, and 4th is peptide bond. So you have to choose the correct answer from the option given below. Quickly answer me in the chat box what you think is the correct option for this question. What do you think, Mohina, Srujan, Rishtiya Mili? What do you think? What would be the correct answer for this question? Okay. Priyadarshini says 1. Chichuni says 1. Okay, okay. Everybody in the chat box. 1. Jury says 1. Okay, okay. Very good, very good. Keep going. It's very easy, beta. They're just asking you the bonds. Okay, let's see. Number 1 is the protein. Protein, which bond? Peptide bond. Remember, the amino acid, they are linked with the peptide bond. Unsaturated fatty acid. I told you that fatty acids are of 2 types, saturated and unsaturated. Saturated single bond, unsaturated double bond. Remember? Swinell says A. Very good. So B. So A is 4th. Okay. B, unsaturated fatty acid. B is 1st. Unsaturated fatty acids are the double bond. I told you. C says nucleic acid. Nucleic acid shows phosphodiester bond. Remember the bond between the phosphate and the sugar, that a phosphodiester bond. So C is 2nd. And D, polysaccharides. Polysaccharide like cellulose, starch, glycogen. What are those? There are polysaccharides, and all polysaccharide shows glycosidic bond. So D is 3. So the correct option will be A4, B1, C2, and D3. So option 1 is the correct option for this question. Is that clear to everyone? Okay, now moving to next one. Which of the following are not secondary metabolites in plant? I told you, remember that table which is given in NCERT, which contains all the examples of secondary metabolite. Please learn that table by heart, beta. Which of the following are not secondary metabolites in plants? Number 1 is morphine, codeine. Number 2 is amino acid, glucose. Number 3 is vinblastic curcumin. Number 4 is rubber and gums. Answer quickly. What you think? Which of the following are not? They're asking not secondary metabolites in plants. Secondary metabolites are those which are not essential for the main metabolic reaction. Everybody in the chat box, Mohina, Trisha, Trujan, what you think? Rishti Amili, what you think? What will be the correct option for this question? Which of the following are not secondary metabolites? Remember secondary metabolites. Swinell says D. Okay. They're asking the not secondary metabolites. Remember this, beta. Primary metabolites are those which are required for the main metabolic reactions, or which play a very important role in the normal physiological reactions of the organism. Everybody answer. Try to participate, everybody. Mohina, Trisha. Vinal says B. Okay. So question says, which of the following are not secondary metabolites? Morphine and codeine, they are alkaloids. They are secondary metabolites. Amino acid and glucose, they are primary metabolites. When blast and curcumin, they are secondary metabolites and they come under the category drugs. Rubber gums, they are also a secondary metabolite. They are polymeric substances. Remember, yesterday you also get a question in the test which says which of the following is not a polymeric substance, right? So remember this table. This table is very important. Rishtia says B. Very good, Rishtia. So moving to the next one. Which of the following biomolecule does have a phosphodiester bond? Very easy. Very easy question. Which of the following biomolecule does have a phosphodiester bond? Quickly answer, everybody. Number 1 says nucleic acid in a nucleotide. Option 2 says fatty acid in a diglyceride. Option 3 says monosaccharides in a polysaccharide. Option 4 says amino acid in a polypeptide. Priyadarshini says 1. Okay, very good, Priyadarshini. Quickly answer, Veda. What you think is the correct option for this question? Jury says 1. Very good, Jury. So these questions as came in NEET. Remember, I've mentioned the year as well. This question came in NEET 2015. So nucleic acid in nucleotide. Remember the bond between the sugar and the phosphate? I told you that phosphodiester bond. The nucleotide shows the phosphodiester bond. So option 1 is the correct option. Fatty acid in a diglyceride. So diglyceride, when it consists of 2 fatty acid and plus 1 glycerol. Triglyceride, if 3 fatty acid, 1 glycerol. Monoglyceride is 1 fatty acid and 1 glycerol. Okay, and the bond is ester linkage. They're ester. Monosaccharide in a polysaccharide, glycosidic. Amino acid in a polypeptide, peptide bond. Remember all the bonds. Moving to next one, match the following again. A says inhibitor of catalytic activity. B possess peptide bond. C cell wall material in fungi. D secondary metabolite. There's options again: ricin, malonate, chitin, collagen. Choose the correct option from the following. Swinell says B. Okay. Rujan, Mohina, Trisha. MS Gauri, Rahul, Ramayana. What do you think? What will be the correct option for this question? Okay, let's see. Inhibitor of catalytic activity. I told you when I was telling you the factors inhibiting the, okay. Moina says 2, okay. MS Gauri says 3, okay. Let's discuss now. So inhibitor of the catalytic activity, I told you the competitive inhibitor. Malonate, remember the competitive inhibitor. Malonate is an inhibitor of enzyme succeeding dehydrogenase. Okay, so correct option for A is to malonate. B, possesses peptide bond. I told you collagen is the protein. Remember, collagen is the abundant protein in animals and rubisco is the abundant protein in the biosphere. So collagen is a protein. And the protein will have peptide bond. So B, fourth. Cell wall material in fungi. Chitin. The cell wall of fungi are made up of chitin. Remember this. E3. These secondary metabolites, resin. Resin is a toxin. Abrin, resin, these are the toxin. Okay, so secondary metabolite D is 1. So A second, B fourth, C 3, and D 1. So correct option is number 2. So all of you who have answered second are absolutely correct and very good all. Now moving to the next one. The essential chemical component of many coenzymes are. I told you when I was telling you about the cofactor. This point is very important. The essential component, chemical component of many coenzymes are. Number 1 is vitamin, number 2 is protein, number 3 is nucleic acid, the fourth is carbohydrate. Quickly answer me in the chat box what you think what will be the correct option for this question. Coenzymes, remember cofactor. Cofactor are of 3 types. Number 1 is prosthetic group. Number 2, coenzyme. Number 3, metal ions. Remember? B. Swinell says B, protein. Okay. Swinell, do you remember that I told that cofactor is a non-protein part and apoenzyme is the protein part? I told you, na? That enzyme, the conjugate enzyme, it consists of two. One is the apoenzyme, which is the protein part, right? And plus the cofactor, which is the non-protein part. And cofactor are of further of 3 type: prosthetic, coenzyme, and metal ions, right? And they collectively form the cofactor plus the apoenzyme. They collectively form holoenzyme, which is biologically active. Remember, I told you. MS Gauri says 1. Very good, MS Gauri. Vitamins, beta, it is vitamins. I remember I told you that coenzymes are those which are temporally bound, to the apoenzyme. Remember this? Which are temporarily bound to the apoenzyme, those are coenzyme. NAD and NADP. They contain vitamin. I told you, and that vitamin is niacin. So the essential chemical component of many coenzymes are vitamin. So option 1 is the correct answer for this question. Protein is not no nucleoside and not carbohydrate. I hope this is clear now. Sunil, now it is clear? Okay, moving to next one. Next question says the primary protein are also called as polypeptide because primary proteins are also called as polypeptide because number one, they are linear chain. Number two, they are polymers of peptide monomers. Successive amino acids are joined by peptide bonds. Fourth, they can assume many conformation. Primary proteins are also called as polypeptide because remember the primary structure of protein? Quickly answer in the chat box what you think is the correct option for this question. The primary protein are also called as polypeptide because they are linear chains, they are polymers of peptide monomers, or they are the successive amino acids are joined by peptide bonds, or they can assume many conformation. What do you think? What will be the correct option for this question? It's a very easy question. It's a very easy question. There's no rocket science in this question. Very easy. Sunil says C. Very good, Sunil. So the question is asking the primary proteins are also called as polypeptide. Remember when this amino acids, they're joined together, like they form a chain, and that chain is polypeptide chain. So the primary structure of the protein shows the polypeptide chain, right? A sequence of amino acid you can see in the primary structure of protein and the polypeptide chain. So the correct option for this question is the successive amino acids are joined together by the peptide bond and they form a polypeptide chain, which is the primary structure. In the secondary structure, you will see the helix and beta sheet. In tertiary structure, you will see the three-dimensional conformation of the protein. In quaternary structure, when more than one polypeptides are present, and these polypeptides are known as subunit in quaternary structure. Moving to the next question, in a reaction catalyzed by an enzyme, which of the following statement is correct? The reaction catalyzed by an enzyme, which of the following statement is correct? They're asking the correct statement. Remember this. Number one, enzyme make transition from substrate to product more difficult. Enzyme increase the activation energy for formation of transition state. Enzyme substrate complex formed during a reaction lasts for a very long time, and the enzyme decrease the activation energy for formation of transition state. What do you think? What will be the correct option for this question? Sunil says B. Increase the enzyme increase the activation energy for the formation of transition state. Okay, MS Gauri says 4. Okay, what about others? Mohina, Trisha, Surjan, Tarun, what do you think will be the correct answer for this question? Okay, so enzyme make the transition from substrate to product more difficult? No, it make easy. Enzyme make the transition from substrate to product easy, not difficult. Enzyme increase the activation energy for formation of transition state? No, it decrease the activation energy. I told you the enzyme increase the rate of reaction by decreasing or lowering the activation energy of the reaction. So enzyme decrease the activation energy for formation of transition state. Third is enzyme substrate complex formed during a reaction lasts for a very long time. No, it lasts for a very short time. Okay, so option 3 says enzyme substrate complex formed during a reaction last for a very long time. No, beta, enzyme substrate complex formed during a reaction last for a very short time. And the enzyme decrease the activation energy for a formation of transition state. This is a correct statement because the question is asking about which of the following statement is correct. So enzyme So option 4 says enzyme decrease the activation energy for formation of transition state. So enzyme will decrease the activation energy because of that the rate of the reaction will increase. I hope it is clear to everyone. Very good, Swinil is D. Now it's already 1. I hope this lecture is informative and interesting at the same time.